Hi, this is Jennifer Bain from Columbia University, here today to speak with you about the natural history study. Thank you so much for having me here today. I wish we could meet in person, and I'm sorry that we won't be able to see you in person, but I hope that I'll be seeing you virtually online this year, and soon enough, we'll be meeting again in person soon. Today, I'd like to talk about the initial description of HNRNPH2 related disorder, and then review what a natural history means. Lastly, I'll present some information about what our group is doing, Simon Searchlight is doing, describe our recent manuscript where we describe 33 individuals, and then talk about how you guys are so important to our collaboration and learning more about this disorder in our virtual platform of data collection this year, and a few introductory slides about other projects and next steps of this exciting research. So, as I mentioned, I'm Jennifer Bain, and I am up in New York City, and Columbia University is located all the way up here, up by the George Washington Bridge, which often gives me lots of traffic problems. So luckily I live in New Jersey, and due to the pandemic, like many of you, I've been working remotely from home in the comfort of my own home with my lovely virtual backgrounds. So I appreciate you all coming to my own home of New Jersey and working with our team from Columbia. As a child neurologist, most of you have probably seen one of us before. You come to the doctor's office explaining all these developmental concerns. And my goal for people who come to the office is to say, well, what is the diagnosis? What do we do to treat this? And when parents say, what can I expect for the future? I want to be able to provide that information. Unfortunately, like many of you probably have seen or noticed, you end up going home with a smattering of different diagnoses like developmental delay, maybe seizures, maybe ADHD or anxiety. And so we don't really know exactly what that means. And in this case, we found out that it might be related to some kind of genetic disorder. But unfortunately, all we have at this point in terms of medicine is really to say we're going to treat this symptomatically, provide therapies to what areas can be amenable to therapy, and maybe medication for some of the symptoms. But darn, it would be great if we had a genetic disorder that we could provide a very targeted treatment to. And I think that's kind of the goal we're talking about here today. What can I expect for the future? If we have a genetic diagnosis, such as HNRNPH2, can we predict what's going to happen in the future? Can we better understand the natural history of this disorder, knowing that we don't necessarily have a targeted treatment? And that's where all of you come in. In the United States, a rare disease is defined as having a condition affecting fewer than 200,000 people in the US. So HNRNPH2 is definitely a rare disease. These rare diseases are often poorly or incompletely understood because the fewer the people, the less we know how to treat them and the less they're usually studied. Most of the treatments, such as drugs or other interventions, require well-designed clinical trial studies that have large numbers of people and that are often replicated in multiple groups. Unfortunately, with rare diseases, these type of studies can be very difficult to run. Therefore, we often end up using a lot of information from something called a natural history study. A natural history study follows a group of people over time who have a specific medical condition or disease. It collects health information in order to understand how that condition develops naturally over time. HNRNPH2 is one such example, where we collect information as individuals get older to better understand what happens in the course of the disorder. It is so important and crucial for rare diseases to have established natural history studies to understand the natural history of them and potentially consider drug development or some kind of intervention and see how that changes or how that shifts the natural history of the disorder. This is a lovely slide that I borrowed from the Combined Brain Initiative that I'll explain in just one second, in which an individual has their genetic disorder identified. They participate in a natural history study, such as what we're talking about today. They speak closely with the researchers and clinicians to say, what exactly does this disorder do to my life, my family's life, and our society? How can we identify things that matter to you and to families and not just what matters to the researchers and the doctors? And how can we use that to better understand what medications or drugs potentially could be safe and effective in treating? Importantly, while all this is going on, you have to work very closely with a whole lot of clinical researchers and basic scientists, such as those who you'll hear from later today, who are studying animal models, cell models, computer mechanisms, biomarkers, and other different model systems to understand more about these genetic disorders and make them potentially amenable to specific clinical trials. Now, the big circle here is kind of talking about what me and my lab are interested in understanding about collecting natural history, understanding what the burden of this disorder is, and really identifying what outcomes matter and what outcomes will change over time. I work closely with the Combined Brain Initiative, and the Yellow Brick Road is one such group that is a participant in this lovely consortium. This is a consortium led by patient advocacy groups. You can see several of them over here, including the Yellow Brick Road Project. And they work with clinicians and researchers and pharmaceutical firms to try to develop treatments for the disorders that each of them represent, and understanding that we are all rare diseases and all rare disease advocacy groups working together, but likely have very similar thoughts and ideals and trial information. So putting together all of our efforts so that we're not recreating the wheel more than once. So why are we here today? We're here to talk about HNRNPH2. In 2016, just 5 years ago, we published a report describing 6 females with variants or typos on the HNRNPH2 gene. This is a gene that's located on the X chromosome. The clinical picture showed a girl, 2 of which are in the upper right-hand corner, with developmental delay and or intellectual disability in the older individuals, some with a diagnosis of autism spectrum disorder, many with muscle weakness and problems with tone, some with seizures, and some with very distinct facial features. When we looked at the structure of the gene, so here's the HNRNPH2 gene, and you can see it's like a long line here. We noticed these 2 arrows. These 2 arrows were where those 2 typos were in the genetic code, and they were seen in all 6 of those girls at the same location. And interestingly, when we looked at where those arrows were causing typos, we noticed that these were very specific instructions that were conserved across all different species. We figured that this was likely a very important spot because of this. And even more interestingly, they were similar typos to the genetic information that was coded in other genes that were also called HNRNPs, called H1 and F. And I'm going to talk about this later. So that year, just a few months later, we met with a group of eager parents at Columbia one Sunday afternoon and came up with a plan to find more families affected by this gene. I appreciate the opportunity to continue working with YBRP and now to Cura Rose Foundation and presenting our research that continues to grow at the 4th annual family meeting. After that time, we also decided to team up with a larger organization called Simon Searchlight. This is the data that I'm going to collect, that I'm going to present with you now, and then I'll present a little bit of our information at Columbia. So what is Simon Searchlight? Some of you might already know about it. The mission of Simon Searchlight is to shed light on neurodevelopmental conditions by collecting high-quality natural history data and build strong partnerships between researchers, industries, and families. The Simon Searchlight group collects detailed medical and behavioral histories along with blood samples. They take all this information, they put it together, and they give it back to families just like we're doing today. They also allow for the free sharing of this data, as well as those blood samples and biosamples, with other qualified researchers. The Simon Searchlight Initiative importantly connects participants and researchers from around the world in order to promote a better understanding of genetic changes that are related to neurodevelopmental disorders. Some of you have already gone through this process. It begins with just signing up online. What's great about Simon Searchlight is that you don't even have to leave your house. You sign up online, you provide your clinical lab report, and then you schedule a medical history phone call with one of the genetic counselors from Searchlight. And then at home, you continue to fill out surveys. If you're able, you can provide a blood sample, and then we continue to follow up with you over time. What's really wonderful is that this is a long-term study, and they gather information from each individual family every year. At the current time, Simon Searchlight has 31 individuals, that's about half of what we have at Columbia, who are signed up with Simon Searchlight. About 24 of them have already started to give their information to Simon Searchlight, and 20 of them have already scheduled that phone call with the genetic counselor. So what I'm going to start presenting to you today is that information on those 20 individuals. 24 people have started filling out surveys, and we do already have some biosamples or blood samples from 9 individuals. We also have a very special research agreement between Simon Searchlight and our natural history study here at Columbia. This allows us to save time and energy on your behalf so that we can share information between both of our research studies. In order to participate and allow this information to be shared, however, you do need to go through the formal consent process for both Simon Searchlight and our Columbia University Natural History Study. By consenting to both of these studies, you allow us the ability to cross the information and to share that information. First, I'm going to present the information from the Simons Registry, and then I'll add a bit more detail from our study where we recently published in Neurology Genetics. The Simons Registry currently has 20 individuals who have completed the full intake and provided their information. As you can see, so importantly, half of the individuals are less than 10 years of age. So we only really have half of our information from little ones. We only have a couple people who are in their adult years. So it's going to be so important to continue finding people who are in adulthood or in adolescence and to continue following these people when they're younger as they're getting older to learn more about what happens as individuals get older. Something that hasn't changed from the original report, we know that all individuals have some degree of developmental delay or intellectual disability. We certainly have learned and continue to learn that language impairment is a very significant problem within the group. 85% report having a significant language impairment. Similar to what we found earlier, about a quarter of the group, 20%, have been diagnosed formally with an autism spectrum disorder. And we do know that other mental health conditions or psychiatric diagnoses like anxiety are also reported in this group. As this is a neurodevelopmental disorder, I am a neurologist and I'm very interested in the neurological conditions that are associated with HNRNPH2 related disorder. Not unsurprisingly, many of the individuals, just like we had first described, have different muscle tone. That's how the muscle is just at rest. So very loose bodies, almost like a floppy baby, is something that many people have recalled being told about their child. 90% have a history of being told they have low muscle tone or a hypotonia. There's a smaller percentage of individuals, these are usually those who are older, who have tight muscle tone, high muscle tone, or spasticity. About half of the group have been told they have a small head size or microcephaly, and many have a movement disorder or report being clumsy. And I'm going to talk about that a little bit more in the future slides. 2 individuals in the group have been given the diagnosis of cerebral palsy, and 1 individual did report having a cyst in the spinal cord. While only 20% did have seizures, we do know that there was a similar percentage who have abnormal brainwave testing on a test called an electroencephalogram or an EEG test. What we've learned is that there's different types of seizures in the group. They can be grand mal, which means full body shaking, petite mal, which is more the absence kind of uh, blank staring seizures, infantile spasms as well. This is a little bit different than the information that we've been collecting on the larger group, but it's good to see that seizures are being reported in this group. Other medical issues that we often see in other neurodevelopmental disorders are things such as gastrointestinal problems. For example, problems with constipation are so common. Problems with reflux or GERD, diarrhea. Eye and vision problems is a significant issue in the group. Individuals have been reporting having strabismus or lazy eye, astigmatism, cortical blindness, or cortical visual impairment. And importantly, many have also reported having orthopedic, joint, or musculoskeletal problems. Endocrinologic problems such as growth problems, difficulty growing, gaining weight, failure to thrive, short stature, and one individual with a low thyroid have also been reported in the group. A few individuals have been diagnosed with a congenital heart disease, though this is less common, and a few individuals less than that have reported having lung problems or breathing issues. One individual also does report having a kidney problem. Simon Searchlight is starting to also ask parents what behaviors are concerning. For example, depression, anxiety, oppositional behavior, aggression, or attention and hyperactivity. We're also studying this a little bit more as these are very important to be intrusive or limiting in an individual's daily life. The child behavior checklist was completed by 9 individuals in the Simon Searchlight group, and things that came up as top concerns include chewing things that aren't edible, sometimes we call that pica, becoming easily frustrated, being afraid to try new things, and also sudden changes in mood. And we see similar things in our group as well. Looking at the Simon Searchlight summary, again, this is 20 individuals. The more common problems that were noted included language impairment, intellectual disability and developmental delay, a low muscle tone, a small head size, orthopedic and joint problems, eye and vision problems, and pica, or eating things that aren't food. Some of the less common issues, but were issues nonetheless, include autism, anxiety, coordination problems, movement problems, endocrinologic problems such as difficulty gaining weight or growing, and heart conditions. What we've realized is that we need to work together. And working together, we've learned more and more about people around the world. And so thanks to all of you, we have really been able to learn more about individuals with the neurodevelopmental disorder associated with HNRNPH2. For those who were not able to read it, we have an open access journal article called The Detailed Clinical and Psychological Phenotype of the X-linked HNRNPH2 Related Neurodevelopmental Disorder. This was published just earlier today and is based on 33 individuals. In the initial paper, we found 2 initial hotspots. I'm going to point them out here, and here, at amino acids 209 and 206. We continue to see these as hotspots in the larger group. Each one of these dots represents a person. The light ones are girls and the dark ones are boys. And yes, you are right. Back in 2016, when I said I didn't think that boys could survive with this, I was wrong. We now have some wonderful boys in our group that are teaching us more about how this disorder affects boys. Today, I'll be presenting on all those individuals, as well as some other individuals with other genetic findings outside of the nuclear localization sequence, outside of these hotspots over here, as well as up here. Now, this is the most updated graph that we have. We have 60 people that are formally enrolled in our natural history study. 43 have already provided medical histories, so we're getting there. As you can see, there seems to be the majority of individuals with those variants at that 206 and 209 variant in that location called the nuclear localization sequence. However, we're starting to learn more about boys, and we're starting to see more cases of siblings and those who are being inherited. Originally, all individuals we had identified did not inherit these gene changes from their parents. More recently, however, you can see up here the triangle, we are identifying individuals who have inherited these changes. So we're starting to learn more about how these genetic changes affect HNRNPH2. It's so important and critical for every single individual out there, whether you're involved in the study or not, to please participate or at least talk to us about your experience. We need to know about everybody so we better understand this gene. As you can see, the facial features we had initially noted continue to be seen in individuals with HNRNPH2. Some of the facial features are even seen in the boys within the group. For a larger picture database, you can definitely check out our newest paper, but here's some of the beautiful faces of HNRNPH2 from around the world. We're also seeing some of those similar findings in hands and feet. We noticed this at the very first family meeting. Long, thin fingers, and for little ones, short, stubby feet that got long and curled out with toes. We've continued to notice that motor skills are a significant concern in the group. Weakness has been reported in many, as well as cerebral palsy, and many have been diagnosed with low muscle tone, and a few with high muscle tone. Coordination difficulties, gait problems, so problems with walking and ambulating, motor planning of our movements have also been noted. The majority of individuals in the group have been receiving physical therapy and occupational therapy, many since a very young age. Now, again, this is a little bit different than the Simons registry I had presented earlier. In our group of more people, we actually have about half of the individuals with clinical seizures and a few more who have abnormal EEG or brainwave tests. It's so important to continue collecting the information from both our natural history study and the Simons group because they are a little bit different right now. Similar though, we do know that the seizure types can be varied. They can be in the setting of fever, they can be staring, shaking, stiffening. The average age of the first seizure was about 8.7 years of age. However, the oldest individual that we do know who had a first-time seizure was an adult at 34 years old. Of those participants who've reported seizures, over half of them have taken an anti-seizure medication, but many of them did stop because the medication was either ineffective, there were side effects, or the seizures went away. Good seizure control has been reported with a medication called levetiracetam or Keppra and valproic acid, also known as Depakote. But we still don't have enough information to say these are the medications you certainly should be giving, and you absolutely should be discussing all of these matters with your clinical neurologist. Again, other things that that matter to individuals. Where are the other intrusive concerns? And again, this is a little bit different than the Simons registry. We found that sleep is definitely a big issue in the group, as in many other neurodevelopmental disorders. We know that autism spectrum disorder likely is in a small percentage of the individuals with this. And so understanding this diagnosis and how to treat this might be slightly differently in this group. Our reports of anxiety actually were much higher in the group, more like 67%. And attention deficit, or at least attention problems and hyperactive problems, was definitely noted. Self-injurious behaviors was noted in a fair percentage of individuals, and most of them were in older people. Similar to the Simons information, we do know that feeding and gastrointestinal problems are very common and present in nearly all participants. Up to half have failure to thrive or growth problems, several even with feeding tubes. Similarly, we found that many have orthopedic or muscular disorders. This could be hip dysplasia in little ones, scoliosis, joint problems of the feet and the ankles. And this is happening up into adulthood. Vision problems are very, very, very high in the group. This is things like strabismus and cortical visual impairment, meaning difficulty in recognizing or understanding vision with the brain. Sensory issues are very common, and I'll talk about that in just one second. And unfortunately, we do know that 3 individuals have passed away with this condition. Many families, I should say, did meet with our wonderful occupational therapy team last summer. Danielle Rowe Martin and Rachel Vogel last year met with about half the families affected by HNRNPH2, and they're continuing to meet with the other half of families who were newly diagnosed or didn't get to meet with her last year. This is what we call a radar plot of sensory processing scores. This is from those sensory profiles that you had filled out a few years ago on 7 individuals. Each individual is a line, and in each one of the corners of this beautiful radar, you can see a different domain. For example, visual processing. An individual that's typical to their peers would be a number 3, so right around the middle. An individual, however, who has much more than others or much more need for visual processing input would be such as here. And so what we're noticing is, unlike their typical peers, different individuals with this disorder have a very atypical sensory profile. They seem to need a lot of visual input, a lot of touching input, and perhaps seem to be very easily affected by movement and body positioning. And I think this is what we need to learn a little bit more about in order to identify how do we help our family members who have sensory processing difficulties and these atypical sensory profiles. If you haven't met the occupational therapy team last year, I encourage you to sign up this year in the natural history study to meet them. One of the biggest questions that many of the young families will ask is about whether their child will walk. The movement team with Rachel Salazar, Sarah Biedners, 2 of our wonderful physical therapists, and our nurse practitioner, Nicole Haluba, have evaluated many of the individuals at family meetings in person as well. We looked at our group in the natural history study, and this shows a group of individuals who achieved the ability to walk. As individuals got older, we did notice that they did have the ability to walk. So individuals still at later ages did have the chance of walking. The probability of being able to walk increased from ages 1 to 6, and then slowly between 9 and 12. By the age of 12, most of the individuals were walking. This is based upon 17 individuals who attended the first 2 family meetings. Again, this is not the entire group, but we did learn that many individuals do gain the skill of walking as they get older, but it often is very delayed compared to typical development. As we learn more about individuals in the group, we tried to modify an 88-point scale to fewer items. Last year, you guys did an amazing job, and we were able to switch to using a virtual platform and still continue collecting information. For those who were here last year, thank you. You were successful in taping 23 small motor tasks, such as these listed here. We're going to continue collecting those same 23 items from those who collected last year and new families who are participating this year. It's critical to better understanding what motor skills are being achieved by individuals and on what trajectory these motor skills are being achieved. And while we were concerned last year that if we shifted our in-person meeting to a virtual meeting, that we would decrease our participation or decrease the enrollment, we actually were able to enroll more families and even reach farther around the globe. You can see with these little purple dots located where we enrolled individuals. In 2017, we were only in the United States. And now, as of just last year, we've really hit most of the continents. So let me assure you that last year, your feedback was so important about the experience that we even were able to publish this. Most of you have received the article. Please feel free to take a look at it. Let me assure you, we want to see you in person. We want to return in person. We hope next year to see you in person again. However, for those who aren't able to join us, we definitely think that utilizing this online platform is important and is critical for rare disease and rare developmental disorder natural history data collection. So thank you for continuing to build this program. For example, in the earlier meetings on the left, you can see that we were working with Doctors Goldman and Damiano Zanato by using a walk sensor with these small little sensors that went into the soles of the shoes of individuals. We were able to validate that and publish that in a biomedical robotics engineering manuscript. And then last year, because you guys did such a great job, we teamed up with Dr. Goldman again and new team member, Dr. Adele Ardalan, to try a new way of collecting data. And what we asked you to do was collect information using home videos and then submit that information using the internet with Dropbox. And in doing that, we were able to show that we could collect very good information. Please keep sending us your videos. We're continuing to learn more about this method of using machine-based learning and hope that this will definitely be something to continue using in the future. I wanted to tell you about a couple of our upcoming studies. One is by one of our recent graduate of the medical school, Gregory Spiropoulos. He is studying the parental experience of genetic treatments. He's interested in understanding from parents like you what your experience is, what providers and researchers need to know in order to better move forward in novel genetic therapies. We'll be sending out a survey, hopefully in the fall, and then he'll be interviewing families as well. We really hope that you'll participate in this project as it hopefully will come around circle and be able to help you and help our team in better understanding how can we treat HNRNPH2 potentially in the future. And after last year's meeting, Dr. Melissa Carter and I have been collaborating to better understand the role of HNRNPH2 and the clinical picture of Rett syndrome. Some people in the group have previously been diagnosed with Rett syndrome. We were interested in learning how this overlaps. Please let me assure you, we are not changing your diagnosis. We are not adding a diagnosis. We're trying to better understand who are other people out there that perhaps have HNRNPH2, and perhaps looking at individuals who have a Rett-like clinical picture might allow us to find more families. So Dr. Carter and one of our other medical students, Nico Gonzalez, will be working on this project. Some of you did receive the questionnaire and complete it already. We already had 24 people reply, and I do hope that you'll be able to reply shortly as well. Tommy Davis is another medical student who's been eager to understand the adaptive and daily living skills of our individuals. He's been working with the physical therapy team, Rachel, Nicole, and Sarah, to better understand what happens to individuals in the group across other domains of living. For example, what things actually matter in the group. What we're starting to learn more about is that there are trends in our overall strengths and weaknesses in individuals. For example, individuals who are walking and individuals who are talking seem to have stronger social skills, stronger responsibility skills, and stronger overall daily activity skills. We're continuing to learn more about this, especially as we learn information every year. We will continue to collect information every year about adaptive and daily living skills to understand more about what happens as time goes by. The last exciting thing I wanted to share about is something I mentioned at the start of the talk. HNRNPH2 is a part of a larger family of proteins. They're almost like cousins, called the HNRNPs. They're designated A through U, and then there's a few other members of the family as well. These proteins are related because they have very similar regions that are denoted by these different colors in the rows on the top. Well, a lot of these have been previously associated with cancers and neurodegenerative conditions like dementia and ALS. We were so suspicious that there might be some overlap between HNRNPH2 and other genes in the family, causing neurodevelopmental disorders. Dr. Maddie Gillentine supported this hypothesis with a recent manuscript entitled Rare Deleterious Mutations of the HNRNP Genes, results in a shared neurodevelopmental disorder. And based on this, Dr. Gillentine has been referring families affected by other genes in the group to our study. I think she's going to be speaking to you later today about this exciting paper. While we are here today to study HNRNPH2, it's also important to recognize that we build power in numbers for potential research, potential interventions, and future studies. I wouldn't be surprised if this will be a learning opportunity for all the families affected by other HNRNPs as well. So we're looking forward to hosting a spring 2022 HNRNP-related neurodevelopmental disorder scientific symposium. Maddie and I will be working on this in the months coming up. And if you have any contacts or any other individuals you think might be interested, please feel free to reach out. How can you help? This is my last slide. Please enroll in the natural history study. I know many of you have joined with us before. I've had many lovely conversations with families, and maybe it wasn't the right time a few years ago. Please do consider that we are always here with open arms to welcome you to our study. Please also consider enrolling in Simon Searchlight. Again, we're not trying to add more burden to your load. We are trying to work together and collect information. Please send us your genetic report and keep answering all these surveys we send. And this summer, we hope you can participate in our virtual data collection by sending videos and signing up for evaluations to meet with me and the occupational therapy team. We like to hear all your new updates. Please send us medical records, MRIs, EEGs, and just keep in touch. Remember, the natural history study is critical. We need to know the good things, we need to know the bad things, and we need to know the ugly things. Everything is important. Every individual is important. HNRNPH2 is associated with a spectrum of individuals. Some are more affected than others, but we need to include everyone to better understand the whole picture. I do have to remind you as a neurologist that seizures can happen. And while we only know a little bit about this right now, we're hoping to learn more about this. Who will have them, when we should be looking for them, and how we should be treating them. More to come on that. And therapies are critical. At this point, we don't have a cure for HNRNPH2, and we don't even know what a cure would be doing. We need to learn more about that, identify what matters to families, and work on therapies, as that seems to be most critical. I have an amazing team of individuals at Columbia University, as well as those who have left Columbia training and have gone elsewhere, but have been enamored by this wonderful group. Remember, nothing in life is to be feared. It is only to be understood. Now is the time to understand more so that we may fear less. I hope that you can thank all of us for being here today. And I really appreciate the opportunity to be here today. I enjoy every year, and I really look forward to meeting with you in person and not speaking to a Zoom seminar anymore. So I hope you have a great time over these 2 days, and I'll talk to you soon. Have a great day. Bye.
Thank you, Dr. Bain, for that update on the HNRNPH2 natural history study and all of the great things happening at Columbia University. Next up, we are going to hear from Dr. Anne Korf, who heads the mouse model research under Dr. Paul Taylor at St. Jude Children's Research Hospital.